Okay, let's come to session 3 of molecular biology. Today we'll discuss a very brief topic, what's nucleoid and what is nucleus. So in our earlier session we learned about what's the nucleus, why we are working with the it's a organized structure where the genome stays or where the genome resides. Okay, and so you have finding with the nucleoid. So nucleoid is we talk in terms of prokaryotes, and nucleus we talk in terms of eukaryotes. Anyway, so the learning outcomes is to remember the variations in the genetic material in prokaryotes and eukaryotes and complexity of nucleus over nucleoid. So what is nucleus? Just understand a very basic thing is nucleus is a genetic control center of a eukaryotic cell. In most cells there is only one nucleus generally. How, of course, you can have two or more nucleus also in some certain clinical conditions, and it has a unique structure and function that is essential to the cell. So if you happen to see the structure of the nucleus, it has a nuclear envelope, okay, which is outside. You have nuclear envelope, then you have chromatin, which is the DNA, and so on. Then you have a small area where you are finding a nucleolus, where you have RNAs and so on, nuclear organizer, what you call. Then you have nucleoplasm. Then you have nucleoplasm is the liquid or the somatic fluid constituent of the nucleus. Okay, then finally you have all the chromatin. Again you have DNA and RNA, right? So there are different areas or different constituents, like nuclear envelope. It's a double layered membrane perforated with the pores. So this is important because when you have a DNA material coming from inside or outside, you need to have a certain kind of pores. Similarly, where you find in cell membranes and so on. So the outer layer is connected with the endoplasmic reticulum, communicating with the cell cytoplasm. So you need to communicate all the transcription factor, all the proteins and so on. It has to come inside the nucleus. Now you see the DNA, where the DNA has to be transcribed. All the transcription translation has to be should be happening in the cell itself, inside the cytoplasm. So the DNA has to come outside, and some transcription factor should go inside. So there, the outer layer is actually important in communicating with the cytoplasm of the cell. Then you have a nucleoplasm. As I was saying, it's a jelly like mostly water within the nucleus. So all the material actually float inside and help the nucleus to keep in the shape. Then you have chromatin. As I was saying, you have a chromatin. The chromatin actually means the DNA and the protein, right? So then there are some regions in the DNA, some regions in the chromatin where the DNA or the proteins are highly dense, what's called heterochromatin region, and there are another regions called euchromatic region. So they have a own importance for their expressions and so on. For example, if you take your eukaryotic euchromatic regions, so the DNA is highly expressed, and heterochromatic region is highly conserved and it doesn't express much. So it has some clinical implications. So the nucleolus is again a non-membrane bound structure composed of proteins and nucleic acids, and so most of the ribosomal RNAs are transcribed into the nucleolus. So this is how the nucleolus, in the nucleus you have certain area called nucleolus where all the ribosomes, ribosomal RNA proteins actually are present. Then you have a different function, like nucleus is actually the command center, right? So main center or master controller, because you have DNA in that, right? So all the DNA which is present in the nucleus, so it is important because it's a residing place for the DNA, which is actually DNA actually commands all the the cell fates and so on. So it plays an important part in creating the cell's proteins, and it's involved in important process dealing with the DNA and other genetic molecules. Of course, you know about deoxyribonucleic acid, where it is present, and also DNA is present in form of proteins and so on. So all this again, nucleus is important in proteins and cell regulation. For example, nucleus oversees cell's function and regulatory mechanisms for keeping the cell healthy and alive, and it regulates in secretion of ribosomes and synthesis of structural proteins and energy metabolism and so on. Then nucleoid is another concept or another structure where it is not much organized. You won't find it, of course, you won't find in eukaryotes. In prokaryotes, it's a region where it contains DNA, and it's not much well organized structure. So okay, the nucleate has an irregular shape, and compared to the nucleus and eukaryotes, which is much circular, and DNA in the nucleoid is much more circular as compared in contrast to where you are finding in the eukaryotes. In eukaryotes you won't find you will find much more organized, but here in the prokaryotes it's much more disorganized. I would not say disorganized in the sense it is more of a not properly has a higher dimensions of packaging sort of thing you're finding in the nucleolite. So as the cell grows, the DNA in the nucleolite may extend into the cytosol and make a cellular fluid. So this is a basic difference what's nucleus and nucleoid I was discussing about. Okay, hopefully you understood the function of nucleus, okay, and the difference between nucleus and nucleolite. Thank you so much.